Greetings and welcome to the CCOF Foundation FSMA Agricultural Production Water Requirements webinar. Thanks for joining us this afternoon. Today's webinar will review the agricultural production water requirements of the Food Safety Modernization Act, also known as FSMA regulations. We will also provide practical tips on how to take water samples and conduct assessments of irrigation water sources. We wanted to acknowledge that today's webinar is supported through a grant from the USDA National Institute of Food and Agriculture. Please note that any opinions, findings, conclusions, or recommendations expressed in this webinar are those of the authors and do not necessarily reflect the view of the U.S. Department of Agriculture. If you're having difficulties connecting to the webinar, if you can't hear, can't see, please call our office for support at 831-423-2263. Press 0 for the operator and someone will help you connect to the webinar. My name is Meaghan Donovan and I will be your host today. I am a program specialist at the CCOF Foundation. The CCOF Foundation advances organic agriculture for a healthy world through education and hardship grants, technical assistance, and consumer education. My colleague Jacob Guth will also be online with us today. Jacob is CCOF's Food Safety Program Manager. Prior to his current role, he worked in CCOF's Organic Certification Department for many years. Jacob will be online to answer questions related to the intersection of food safety and organic. Before we get started with the main presentation, I wanted to review a few housekeeping items. We are happy to announce that this live webinar broadcast has been approved for Certified Crop Advisor Continuing Education Units. To receive credit for attending the webinar, include your full name and Certified Crop Advisor certification number in the evaluation form at the end of the webinar, and you must attend the entire webinar to receive credit. I also wanted to let you know how you can participate in today's web event. We're looking at an example of the GoToWebinar attendee interface, which is made up of two parts. The viewer window on the left, which allows you to see everything the presenter will share on the screen, and the control panel on the right. Within the control panel is how you can participate in today's event, so let's look at that. By clicking on the orange arrow, you can open and close your control panel. From the View menu, you can also set the control panel not to auto-hide when inactive if you prefer to keep it always open. The audio pane provides audio information. By default, you have joined the webinar via mic and speakers. If you prefer, you can join the audio via telephone by selecting phone call, and the dial-in information will be displayed. During the presentation, you can send questions to our webinar staff through the questions pane. Simply type in your question and click send. Throughout the webinar, we'll stop from time to time for questions and answers. As a final reminder, today's webinar is being recorded, and everyone will receive an email with a link to view the recording of today's event, along with a PDF of the PowerPoint slides. Today, we also have a copy of the PDF of the PowerPoint slides available for download through GoToWebinar. Click on the blue file name in your handout pane. The PDF of the slides will open in your internet browser. You can view the slides in the browser or download them onto your computer. To expand or collapse the handouts or questions pane, click on the triangle next to the name of the pane. For example, if you can't see the box in the questions pane, click on the triangle there and the pane will expand so you can see the full box. So we wanted to encourage you to check out or test out the questions pane. Let us know what crops you grow and if you currently test your irrigation water for E. coli. So write us in, let us know, and we'll loop back around to that information a little later in the presentation. I also wanted to note we had a lot of people sign up for this webinar, which is really great. So we're going to have a great crowd in the audience today, and we'll try to respond to as many questions and comments as possible. We wanted to apologize in advance if we can't get to your specific question and encourage you to follow up after the webinar. So I also wanted to give a little overview of how this webinar fits into the broader context of the Food Safety Modernization Act. And so please note today's webinar is for farms that must fully comply with the FSMA Produce Safety Rule, and farms fall into this category if the average of the most 3 years of gross sales from the farm is greater than $500,000 in 2011 dollar equivalent, or if the farm sold less than $500,000 adjusted average gross sales, but did not sell more than 50% to a qualified end user, such as a restaurant or local store or individual. So if your farm has been working on FSMA compliance for a while, you may know if you're fully subject to the rule or not. If this is your first introduction to FSMA, you might be saying, that was super confusing. I'm not sure what you just said. I need more information to figure out what you're talking about there. And because FSMA can be kind of confusing, last year the CCOF Foundation and Community Alliance of Family Farmers partnered to host a series on FSMA for organic produce growers and professionals. And so if you're still trying to figure out if you're fully subject to the rule or not, I encourage you to check out the How Do I Need to Comply with FSMA webinar. That walks through all of the different compliance levels and helps farms figure out where they fall, whether they're fully subject or they fall into some of the exempt categories that exist for different reasons. It may be that your annual produce sales is very low, so you fall into one of those exempt categories, or there are some crops that are not subject to the produce safety rule, such as potatoes, because they're not commonly consumed raw. So there's a lot of different nuance and details. So if you're still trying to figure out where you fall, check out that first webinar there. The second two webinars go into detail of all the requirements farms need to comply with if they are fully subject to the rule. And this is just a reminder about what all of those are. But for today's webinar, we are going to focus specifically on the agricultural production water requirements under FSMA. And so we're going to do a deep dive, no pun intended, into those requirements. And just note that the produce safety rule splits out harvest and post-water requirements, and those are different, and we will not be going into those today. We're just focusing on the production water requirements. So with that, I'd like to introduce our speaker. Ronald Bond serves as a water quality researcher at the Western Center for Food Safety in the Atwell Water and Foodborne Disease Laboratory at the University of California, Davis. His current research focuses on measuring the variability of microbial water quality in agricultural systems. Through his work, he collaborates with produce grower associations, irrigation districts, and individual growers to conduct research and outreach on best practices for compliance with the agricultural water requirements of the Produce Safety Rule, as well as ways that growers can, as well as ease the burden of the requirements for growers. So, Ronnie, thank you for being here today, and I'll pass the presentation over to you.
Thank you, Megan. Appreciate it. Hi, everyone, and thank you to the CCOF for hosting this today. These agricultural water requirements are important, I think, through FSMA, and we'll get into why they are important. The first thing I want to talk about was, one is who I am and who I work for. I work for two organizations within UC Davis. One is WIFS, and maybe you've heard of that, and the other is WCFS. And I get this question all the time is, well, what's the difference between the two? Well, WIFS is the Western Institute for Food Safety and Security, and it's a research institute based within the School of Veterinary Medicine and the College of Ag and Environmental Sciences at UC Davis. It's a collaboration. It's mostly engaged in education, outreach, and research in areas of food safety. Well, not only areas of food safety and water quality, but also bio food safety or bioterrorism and other things like that. Our funding is diverse through WIFs. We can get county, state, and federal sources of money. The Western Center for Food Safety is an FDA Center of Excellence. We mainly focus research on, well, currently FSMA implementation. We are 100% funded through the FDA, so no state or county or local funding sources for those. So that's just the small, the differences between the two. I get this question all the time, so we think it's important to caveat that. So know who to contact for which expertise there. Okay, so what we're going to talk about today, we'll go into a little bit of regulatory water microbiology, not a deep dive into microbiology, or at least water microbiology, but enough to get you kind of thinking about why regulators regulate the way they do. Then move into specifically the ag water requirements under FSMA, and then some practical aseptic water sampling, how to sample and how best to sample. And then into the environmental assessment, a portion of FSMA, the annual inspection of your water system, and then a little bit of talk about research. So why study microorganisms in water? Well, water is not only essential for our health, or human health, but it's also essential for the health of microorganisms, and they thrive in it. They thrive in it whether it's fresh water, hypersaline, hot, cold. So different types of environments, microorganisms are pretty comfortable in water. And there are tons of ecological processes that both either degrade or let them grow during their life cycle. And some of the areas of research are focused on a lot of, especially with surface water, that most ag water is, especially in the West, is UV sunlight, inactivation of microorganisms and how that plays a role. Also temperatures and interspecies competition. One goes in the other, and then like biofilming and things like that, and how long they persist in the environment is where research is heading to better inform these regulations. So why test for bacteria? Well, they provide a pretty good baseline for water quality and really ensure the public trust. So it's akin to beach monitoring, where if you see a sign out on the beach, you're likely not going to swim at that beach because, well, it's contaminated. So, and then why we monitor for pathogens? I'm sorry, for bacteria. Well, indicator bacteria, we monitor for indicator bacteria pathogens, and there are some promising technologies for host-specific bacteria like Bacteriales, where you have a contaminated a host like a deer or cow or cattle or pigs shedding these bacteria. Well, we have technologies that can actually quantify that, and they're a little expensive, just like the pathogens, and they're not as in large numbers as indicator bacteria, but we'll get into that here in a second. So the historic transition, looking at indicator bacteria, which is a standard measure for most public health, has mainly been focused on a singular group of bacteria, the coliform group, specifically the total coliform groups, about 100 years ago or more. Lately, recently, fecal coliforms were the standard, and now increasingly E. coli has become the standard for measuring public health water. Or waters for public health. And fecal coliforms are pretty abundant in feces, which the name would imply. And E. coli, while not as abundant, are still fairly abundant in those, and a little more protective than the fecal coliforms. At least that's what the science is saying. Now, Enterococcus, we're not going to really get into today because it's not a part of FSMA. But it's a good idea to know it exists. It's actually in a completely different family, and they can cause illness, and they're very persistent in the environment, much more so than E. coli. So the major waterborne pathogens of concern are, when we talk about pathogens, we can mean a number of things. We can mean bacteria, viruses, or protozoa. For bacteria, pathogenic E. coli has gotten a lot of headlines, especially E. coli 0157H7, which is Shiga-toxin producing E. coli, or an Aztec. And even some non-0157 Aztecs have gotten some attention, such as like 0145 or 026, that you probably heard of them. And you certainly heard of Salmonella or Listeria. Listeria more so in post-harvest environments, but they're also pretty persistent in harvest environments as well, or pre-harvest environments, specifically monocytogenes, Shigella. And then for viruses, hepatitis A and norovirus. Norovirus is the one that gets the most headlines at this point. It not only makes you sick, it's actually pretty quick actor on your body, and it can actually be passed like from a doorknob. You know, someone contaminates that doorknob, you touch it, you may get that disease. So it's not only just foodborne, food contact, it also is person to person. And some protozoa like Cryptosporidium, Giardia, and Cyclospora. So now that we understand what are indicators and what we mean by pathogens, we need to understand why we test for one as an indicator of the other. Well, that's because ideally, indicator bacteria actually tell us something about the persistence of pathogens. The graph on the left shows a one-to-one relationship between indicators and pathogens. So if we test for one, we understand the risk of the other. So the graph on the right, we have what we call a threshold phenomenon, where we start out with pretty much no relationship, where you have pathogens the same as indicators. Indicators are the dotted line. Sorry, pathogens are the round circles in there. And until there's an actual threshold, where meaning that once we cross that threshold, where there's a sharp increase in indicators, we have a large increase in pathogens as well. So that's the ideal world. So that's the assumption. So that, I'm sorry, let me go back one. Let me go back, sorry. I'm almost spoiler alert there. So that's the assumption that under which current regulation exists. So this threshold value. So under FSMA, it's the 126, and in the real world, we don't see that relationship. In fact, these don't act the same as because of so many inputs in the farm environment. We have a lot of inputs. So you have a runoff from some operation. You could have wildlife inputs, human inputs. There's a number of inputs. So there's so much going into a waterway at any given point that it's hard to correlate pathogens with indicator E. coli. And then again, with the threshold phenomenon, it's the same idea, that the threshold may not meet or may not be what the regulatory community sets as a threshold, may not be far out enough to catch that increase in pathogens. So at 126, you may need a threshold at 10,000 to catch that, I don't know, a sewage spill or something like that. So that's what we mean by the threshold phenomenon and why these things don't really correlate to one another, but they're a good idea to test for anyway, because it does ensure public health to some extent. So how do we actually measure for bacteria in water? Well, we take a sample, we bring it to the lab. It's filtered on these specialized membranes, so paper-thin little membranes. These membranes are then placed on specialized media. In this case, it's modified MTEC, and all that means is method 1603, the same method that is used or is cited in FSMA. Then it's incubated for a certain amount of time, so 2 hours at 35 C, 44.5 C. And then after 24 hours, the plates are taken out of the sample. And I should say that you're filtering 100 mils in each of these. So let's just say everything's good, you're filtering 100 mils. And then once it's counted, that's reported to you, the grower, either by your lab, if you have in-house lab, or someone else testing your water for you, that's tested. So in this plate, you get an 80 CFU, which is colony forming unit. I'll explain that in a second. Colony forming unit per 100 mil. So what to do with those results? Well, you probably will get a couple of, there's two types of results basically from a microbiological sample. One is what we call an MPN, or most probable number. And it's a result based on the likelihood of testing positive. So if you look at the illustration below, I'm not going to go into the method, but that one sample is diluted times 5 times 3. So the result is 4 tubes at the bottom tested positive, 2 tubes tested positive, and this is the same sample, by the way, tested positive, and 1 tested positive. So what the lab does is that's basically a number, so 4, 2, 1. That 4, 2, 1 is then, you look at a chart, or you have a MPN calculator on your computer, and it spits out a number, and it gives you an estimate of how many bacteria are in that sample. Now, CFU, or colony forming unit, is a direct count. So it's a single sample run on a single volume, just like you saw in the illustration before this, the slide before this. And that sample result is counted from a single plate, just like you saw the 80 CFU there. One volume's filtered, and the other is, the result is given to you like that. Now, neither one gives you actual number of bacteria, because colonies have tons of bacteria that form that colony. And an MPN is grown up in what we call an enrichment. So we basically take a sample and let it grow all night, and then you get a positive tube or you don't. So they don't actually give you the number of bacteria. It's an estimate based on that criteria. So what are the natural routes of contamination? How does feces get to the consumer? Well, a number of ways, especially naturally. It can be soil, water, plants and animals, equipment and humans, and it moves right through the system pretty easily, pretty rapidly. Now, waterborne routes on farms are the same thing. They have the same type of movement. It goes from contaminated water. It can contaminate the produce. The equipment can be contaminated. Humans can actually be contaminated with contaminated water. And also, humans can contaminate equipment, and equipment contaminate produce. So it's a whole mess that has to be managed. And you guys are all aware of this. It's tough to manage everything all at once. So understanding this system is important. So we have a lot of, not just published this, we published this a while back. It's an introduction to waterborne pathogens in agricultural watersheds. It's basically, our source material is based on current microbiological methods and best practices. It was developed for practical on-farm education of pathogens, and it's all forms of pathogens in all form of agriculture. And it was based on the NRCS's wish to start reducing on-farm contamination, to get this message out there. and it contains tons of references. So if you have a chance to look at this, I think we may include it in the handout at the end of this. But you should take a look at this. It's a good reference material.
Great, thank you, Ronnie, for the introduction there. And I just wanted to open it up to questions from the audience. So if you have any questions on the overview, your crash course in microbiology, feel free to write those in. And up next, we're going to be going more into the FSMA regulations, so we'll be getting to that type of content just next. To loop back around about who we have out in the audience today, we have growers that are growing almonds, table grapes, stone fruit, avocados, lemons, veggies, alfalfa, strawberries, and edible flowers. So diversity of growers out there. And then most respondents are testing their irrigation water, and several of them specifically mentioned E. coli. And then there are a few folks online who aren't testing currently, but planning to do so. So a question came in, and they're wanting to know, what questions should I be asking my lab to be sure that I'm testing to FSMA standards?
That's a good question. We're going to get into that, actually, what for FSMA standards. We'll get into that in the next part. I don't want to spoil it, but testing definitely for E. coli at this point. Whatever method that they're using, it's probably going to be accepted, because you'll see here in a little bit, the FDA released a pretty exhaustive list of methods, and most and you'll see that on there. So that's something that'll be answered in the next, I think in the next part.
Great, thank you. And then we had one more question that came in. So if someone's asking if their water is not up to standard, how do you clean that water up?
Not up to standard. I would hesitate to say that you could clean it. It depends on the water source, so which one it is. I think that's a good question. How do you clean it up? You can treat your water. The FDA has come out with microbial die-off thresholds. So you have you could wait to harvest post irrigation, and that's a way to get around a high count. But I've tested a lot of water in California and in Washington State now for the last 4 years since FSMA came out, and a lot of them are high. They're definitely high in California, higher than in Washington State, and they all could comply. It's just you have to take the water quality profile as a whole and see if that is something. Your 1 to 2 samples, I think you're probably taking maybe for GAPS compliance or something like that. You'll want to look at over the years, do all those samples actually pop the threshold? And you need to look really, and we have a calculator on the Western Center website, so wcfs.ucdavis.edu, to calculate your water quality profile. And it doesn't have to be the 5 and 20. You can just take results that you've gotten, at least 20 over the last whatever years it is, and plug them in there and see if you actually pop that statistical threshold. And I'll tell you about that in the next part.
Great. So just a couple of follow-up questions on the cleaning up the water. People were wondering whether you recommend using chlorine or disinfect with peracetic acid.
Chlorine is pretty sticky. I mean, it depends. So in surface water supplies, I generally don't see that as a cleaning. A lot of folks will irrigate out of ponds, and they use bluestone. and it's an effective algicide, also an effective bactericide. So I would say chlorine. I mean, if you have that in line and it's safe and you can use it, certainly I don't have any problem with it. I don't see any problem with it, especially if it doesn't harm the crop or the environment. So that I think I've answered that, maybe.
All right, thank you. And just a reminder too, before you do anything to use any products on your farm, always run them by your organic certifier, just to make sure that they're allowed for use in organic, and yeah, that your certifier is in the loop about that. So we're going to take one more question and then move on to the next part of our presentation. So someone wrote in wondering if the test is not a good indicator for contamination, why is it required?
Good question. That's the number one question we get, because there's nothing else. Nothing else that comes to that standard of public health protection. The EPA, and before that, I can't remember, it's something public health, has done an exhaustive epidemiological studies over the years, from going from the 1920s all the way till now, looking at these standards and updating these standards. Now, if FSMA is around in 50 years from now, will we see a better standard? Probably, than E. coli, but they're the best public health protection right now. So there are other technologies, like I said, coming online, but they have to get cheaper, because growers are not going to pay for a genetic test, because it probably would be burdensome. No, it would be burdensome on the grower. So I would say that it's easier. It's an easier test. It's a cheaper test, and at certain thresholds, which we were talking about, it's an effective test.
Great. Well, thank you. So we're going to wrap up questions for now, but we'll have a couple other questions break throughout the presentation. So if you think of something else you want to ask, send it in during the next half of the presentation, and we'll loop back around then. So back to you.
Back to me. Okay. Oh, I went too far. Okay, now we're going to talk about FSMA Subpart E, which is the agricultural water requirements under FSMA. It includes, but not limited to, the definition of ag water, the testing requirements, the criteria for those testing requirements, handling exceedances, just like the question before. What do I do if there's an exceedance? Environmental assessment or annual inspection, which go along with ag water, and data sharing, which I'll talk about. We're doing a little bit of research on data sharing to help reduce the burden on growers. So we'll talk a little more about that later on. And then compliance dates. So what do we mean, or what does the FDA mean by agricultural water, or pre-harvest or production water? In the definition, it says, in part, as water that is intended to or likely to contact the harvestable portion of covered produce or contact surfaces, or food contact surfaces. Now, that means that you have a sprinkler, your overhead sprinkling lettuce. Lettuce is easy because everyone sees lettuce everywhere in California. It's overhead. You're probably gonna have to comply, unless you're irrigating with municipal water. Now, farms are super complicated, as I know, testing all these waterways near farms and actually on-farm testing that we're doing. You have all types of mechanisms, water delivery systems in place, and it is sort of, you have to actually think about, well, do I need to comply? Because, you know, I might have oranges on a drip system, but I'm using surface water or canal water for fumigation spray. Well, you'll have to comply at that point. You may have to comply. You could change that over to a municipal source and you won't have to comply. Apples on a microjet system. but they're, they're on a microjet sprinkler system. Maybe they're on groundwater, which you'd have to comply anyway, especially if you're talking about apples on a trellis versus apples on a tree, a traditional orchard. But your overhead cooling using a treated bluestone pond water, yep, you're going to have to comply. And then you're going to have to find the source of that water. One, you have two sources there, and we'll talk about that later. And under FSMA, it's up to the grower to document the reasons for noncompliance. It's up to you to decide whether or not you need to comply or you are exempt. So some examples of agricultural water can be canal water, sprinkler water, however it's conveyed, groundwater, pond or reservoir, spray application, and overhead cooling or overhead sprinkling. So what are the standards? What are the criteria? So the criteria are complex because the nature of water is complex. I think Samir Ansar said that, and I liked it, so I repeat it. He's with FDA. So what you actually need to test for? Well, you need to test for E. coli. Like the question before is, what do I ask my lab? Well, do you test for E. coli? If they say yes, if they test water specifically for E. coli, you say, okay, great, test my water for E. coli. The threshold, which we talked about, these thresholds for E. coli is a geometric mean, or GM, not to exceed 126 CFU. And I should say the GM, CFU value they've also added in an update the MPN a year ago. So you can use, your lab can test, they can use CFU methods or they can use MPN methods. And then the statistical threshold value, not to exceed 410 CFU per 100 mil. If you want to know more about that, we can get that in the question and answer section. What's the difference between GM and statistical threshold value? But just know that when you get a result, your lab is going to give you a result per 100 mil. So you want to know what that result is per 100 mil, and then see where you fall within that criteria. Now, how often you sample? The size of your profile, what we call a water quality profile, depends, or what they call a water quality profile, depends on your water source. If you're mainly on well water, you'll have to take 4 initial samples, and then 1 sample a year after that for up to 4 years. And then I think it's after every 10 years you have to reestablish that water quality profile. So for surface water samples, it's 20 samples, 5 samples a year for 4 years, and you have 2 to 4 years to complete these water quality profiles. So you can complete them early in 2 years, or you can just take your 5 samples every year for 4 years. When do you sample? Well, as close to harvest as practical. Now, I know that's an open-ended word, but generally it means that for long harvest seasons, you'll want to spread out your samples, and again, for short seasons, you'll probably have them close together. That's okay. Those 5 samples a year, or the 1 sample. And then for multiple harvest seasons, it's 1 profile for each. And where do you sample? Well, we're going to get into what is a source, but there's no specific location required, but it must be representative of your source. If you're taking from a canal, you might either want to take it from the turnout or the turnout near your farm. So however if it's a piped turnout to your farm, you'll want to take it there, as close to your field. I mean, you don't want to take one 2 miles away if the canal is 2 miles away. So however that water is conveyed to you, take it as close to the source as possible. And it must be consistent over the course of your water quality profile. So that site you sample, the 5 times should be the same site every time. Now, FDA is currently working on simplifying the ag water requirements under E, with recommendations and guidance coming out all the time. So it's a good idea. One, if you haven't read the FSMA Subpart E, the comment section, and everything that has to do with that in the Federal Register, it's actually it's pretty informative. They do an exhaustive comment period, and they go through each of them with pretty good answers on all these questions you're probably having and why they actually did what they did, came out with what they came out with. So why are they picking on water? Well, contamination can't move from point to point without a vehicle, and water is a great vehicle for transporting pathogens. I mean, in Yuma, Arizona, they're dealing with that. Somehow, irrigation water was contaminated, and they think it was the CAFO that was nearby, but how did that get to the field? That's a good question. And why did it persist so long? So they're dealing with all these complex questions and trying to get to the answer. Well, they come out with new metrics to say, well, you need to do something with that water if you're near that type of operation. And then for an outbreak to occur, there must be multiple pieces of produce contaminated, and water is a perfect way to broadcast contamination through a field. So one little turnout, if it's just pulling in contamination, can broadcast an entire block through an entire block of produce. And also, bacteria must survive long enough. So, in many studies we've shown, and others have shown, Cornell is currently doing this, and CPS-funded research is currently looking at these increased survival rates once there's an irrigation event in bacteria on produce. And it shows, I mean, typically shows that they have a long persistence period post-irrigation. But the goal of most food safety programs is to reduce this risk. So knowing all this, you can go out, sort of mitigate these risks and reduce them altogether. So when do you need to comply? Again, these dates changed. There was a 2017 update from the FDA that said we're going to take a look or simplify these rules. So we're going to change when you need to actually start complying. So for very small farms, it was at the time 8 years, now 7 years to comply, 6 and 5 for the other categories. Now, along with that, and we'll talk about this later, the environmental assessments, they need to start as soon as you can, really. You need to start looking at your water, your source, your delivery, and all those things. So we'll talk about how to do that here in a little bit. Another big update from the FDA was, again, these multiple testing methods. Originally, it was only method 1603, EPA method 1603, which is an E. coli test. It's used in freshwater systems and used to be used in marine systems as well, but they've accepted a number of acceptable testing methods, because what they realize is that requiring one method that a lot of testing labs don't actually conduct was burdensome on the grower. So they pushed these other methods out immediately as a way to sort of mitigate that concern from the growers. And I thought it was a good thing. But there's one thing to remember about these methods is, while it's not in the rule, it's probably a good idea to have these samples run by a certified lab. And what I mean by certified is that they have some either ISO or ANSI, ANSI certification to run samples, whether it's your county lab or any of your corporate testing labs. So implementing this rule is going to be a monumental feat. I mean, we California, have over 45,000 farms listed as far as the last agricultural census, with 85 billion in production. So it's monumental in California, and I can't imagine what it is in other states as well. I mean, or nationwide at this point. But we do have food safety. California is coming out all the time with new food safety programs, like the Produce Safety Program through the CDFA, through the California Department of Food and Agriculture, and then industry and private groups like the CCOF, LGMA, Safe Food Alliance, the Produce Safety Alliance. All these guys are coming out with educational materials to really get farmers up and going and not be left behind. We do recommend that you start at your local office supply store, grabbing 3-ring binders. You're going to need them. You're going to need to document everything. So definitely getting a head start on that is probably a good idea. I don't know, at 45,000 farms, it's going to be a lot of 3-ring binders. And the same is true for other states. So it's important to reach out to your state departments of ag or groups like that, like the Certified Organic Farmers or again, the Produce Safety Alliance for advice if you need advice on this type of thing, on implementing. So now we're going to get into the more practical, less about the regulatory, or not about the regulatory, and more into the practical of aseptic water sampling, how to actually take a water sample. It involves sampling in a way where you avoid contaminating your sample by the method you use. So don't put your finger in it, basically. Don't become the sample. That's where we get in trouble when we test water, or where I take a person out, whoever it is, and they take a sample and their hand goes right into the sample, not thinking about the consequences of that. Where you start when you're thinking about water sampling is you start at your source. Again, you need to be as close to your source as practical through FSMA, and not all sources are created equally. Like illustrated here, you have a piped water system with sand filters. So where do I sample? Well, you'll want to sample either post or either before or after the sand filters. I would say after, because it's a filtration system, so it's probably going to be more representative of the water you're conveying to the produce. Also, like this surface water system here, this little lateral canal where there's a turnout there that goes out to a produce farm, that's probably going to be the best spot because there's no, at this point, there was no way to sample the pipe system because there was no spigot in place. Or something like this lined reservoir, this lined pond. Where to sample in a pond depends on where you're actually pulling the water from the pond to do whatever type of irrigation you're going to do, whether it's overhead cool or just natural or watering. So here's an example of a complicated system. You have on the right at number 3, you have a canal coming down, flow is heading towards you, and there's a turnout. You can't see it, but there's a turnout there, a gate turnout, and it flows over across the road and down to that number 2. That's a little turnout there that will that can in and of itself be used to irrigate the field. And then that is split to the field, and it runs to the pond there, and the pond is filled with that irrigation water, with the surface water from the canal. So choosing your sample point, is it site number 3, site 2, or which one of the number 1s do you actually sample? What's complicated? It depends on what you're using as close to harvest. So you'll want to understand that better, and we'll explain how to actually do that in the environmental assessment portion of that. So in my suggestion, if you're overhead cooling with that pond, that's pretty much where you'll be sampling. But during the season, these were apples. During the season, they're mainly going from the canal water, so they're only using that pond to overhead cool and later in the season. So it depends on when you're actually sampling. But I should point out, before I move on, I should point out that each one of those is a source, a potential source. You may actually have to sample all 3. It depends on how you're using the water. So again, choosing the sample point it depends on the source, obviously, but there are some surface water considerations you should keep in mind. Is basically, where is it easy to take a sample? Is it a bridge? Is it a turnout? Can you sample at your turnout? Can you sample at your pipe turnout on your field? That's something to consider. Also, how far does the water travel before delivery? You'll want to consider that as you're taking that sample. And is the water filtered prior to reaching delivery? Like I said in that the slide, 2 slides before, is that the filter is important because it actually does its job, even if it's a mesh filter or a sand filter. And is there any canal maintenance going on? You want to consider that. Irrigation districts maintain their canals constantly. So, oh, sorry, I advanced it too far. Sorry, guys, I lost my, oh gosh. We had some built-in movement in this thing. Are they maintaining the canal? Well, you'll want to know that. And if you don't have a relationship with your irrigation district, you probably should at this point. So some considerations for piped water. You see the photo on the right shows these big mesh filters in line there. Again, they're effective. So where do you actually sample, or what are the things to consider? Well, is there a clear point before the point of application? Do you have a spigot or a valve in place? If you don't, you should probably get one put in. Is there any backflow prevention? We'll talk more about backflow prevention, but that's something FDA is keen on looking for when they're on your property. And can the sample point be cleaned easily? It's important that you're able to clean your sample point, whether it's a spigot or a valve or any type of any way to actually pull water from a piped water system. And now irrigation, active irrigation isn't necessarily a bad thing, but if it's under pressure, then you're going to be shooting water a pretty good distance. So taking a sample from that is pretty difficult. So you'll either get wet or some other disaster. So you'll want to actually put in some kind of either a barb or something like that to actually collect that water sample. And avoid the green manky hose. We say this everywhere. We see professionals from testing labs take water samples from the hose, and that's a big no-no. You want to pull that hose off of that spigot and sample from the spigot, because the hose is full of biofilms and full of everything else. You'll want to definitely take that off. Avoid that. If you can bring your own hose, if you actually need to sample from a hose, then that's a better idea. Bring your own clean hose, but don't avoid sampling from the green manky hose. Now, FSMA requires that there be no foreseeable contamination between the source and delivery. So if you're going to pick a site at your piped water, or your piped site at your field, you'll want to make sure there's no point source. There's no contamination between there. So what that means is if you're taking from a lateral and you pull that into a pipe system, that you don't have a break and there's, I don't know, cattle manure spray going over in the next field. You just want to make sure there's no reasonably foreseeable contamination between you and the delivery. And I know it's hard to ensure that sample points are consistent. We say that all the time, that different risks associated with different sample points, like piped water system, surface system versus groundwater. Groundwater is a pretty clean system, should be very clean system. When you're looking the difference between piped surface water and piped groundwater, they're different. So there's different risks associated with them. So you should be aware of that. And then, as far as FSMA, you really have to show the inspectors where you actually sample. So knowing where you sample and not saying, well, I think it's on the corner of X and X. Well, you actually have to know where you actually sample. So move on from there. So what you need to actually take a sample? Well, you definitely need clean gloves. I don't mean sterile gloves. Aseptic sampling doesn't involve sterile gloves. You need clean gloves. You need a sample bottle. You might need a sampling pole, especially if you're sampling from a canal. And you need we request that you use alcohol pads. That's to wipe down piped water systems. If you have a spigot, you can wipe it down. I'll also explain more of that on the practical aspects of it. And you need a cooler. Now, it's not necessary to have a cooler with wet ice or blue ice. I mean, it's not necessary. It's necessary to have a cooler with ice. either wet or blue ice. Now, as far as the sample bottle, you need a couple of things. You need labels on the bottle, and they need to include an ID, a time and date, where you are, the initials of your sampler, and we won't get into sodium thiosulfate because if you're using municipal water, that's probably what you'll need, but I think most of you probably are not using that. If you are, we'll get that in the question and answer section. So here's some practical tips. So one, don't overcomplicate it. And be prepared before you begin. And use the aseptic technique, so not putting your finger in it, not setting it down or anything like that. Taking care of your sample all the way through, sampling. Labeling clearly. Keep the labels clearly. Somebody that doesn't write with their foot is the best idea for labeling. That way you know that the lab can copy your sample location ID and all that clearly. And document everything. FSMA requires that you document everything. If something happens, document it. Protect your samples all the way to the lab. You should protect your samples all the way to the lab. That means once you take the sample, cap them, put them in the cooler, just as quickly as you can. And then record results clearly. Again, keep everything clear, written down in your binder or on your field sheet. Now, don't forget something to write on. That's a good habit. You should always have a piece of paper, always in the truck or the car, or however you collect your samples. And you should develop good habits, because keeping track of time is an important thing. You know, you got to get these samples to the lab at a certain time. So it's important to do that. Mark your sites with GPS. We recommend that. Even using your phone to mark that down and then record that later on your sheet is a good idea. You should always have some type of label with you, whether it's duct tape or the labels that a lab will provide you to write on. And you should make note of anything noteworthy. Is there bird feces on the turnout that I'm sampling from? Are there ground squirrel nests nearby? Is there fecal material on the side of the canal, which we'll talk about here in a little bit? Is there a deer carcass floating by? I mean, that happens. And does anything need repair? So you should mark that too. In your pipe water systems, do you need to repair things? So here are some best practices. The first thing we do when we start a project is we make pre-trips. We go on pre-trips, so reconnaissance trips, and practice sampling. You should already be practicing. If you're not already sampling, you should start sampling, start practicing sampling. You should glove up. Glove up is not going to protect you from contaminating your sample, but it ensures that you're ready to sample. So I got my gloves on, I'm ready to sample. Keep the bottle secure. Keep the cap secure. I can't tell you this enough. I see more people contaminate their sample by touching the inside of the cap. And so it's an invalid sample at that point. So let me show you an illustration here of how we actually take a sample. So I've got the bottle neck down, facing down. And this is one swift motion, but I'm gonna go through it in chunks here. I plunge the bottle neck down into the water about 6 inches below the surface, move it through the water column, facing up. You see the air coming through, and then on up. Now, that water I'm pulling out is because I filled the bottle too much, so you need to ensure that it has enough headspace, and we'll explain what headspace is here in a little bit. Ensure that there's enough headspace so the lab can homogenize the sample, basically shake up the sample, because if it's too full, they can't shake the sample. So again, for pipe water systems, some of that same stuff apply. Some of the same rules apply, except that you're sampling from a spigot or a sample point at a pipe system. You'll want to open that system up as soon as you get there. Just blast it. Open it all the way up. Blast it for 2 to 5 minutes. Once you stop seeing turbidity or any rusting or anything else going on, you'll want to shut that off and then grab an alcohol swab, which you can get in any med kit, and take and clean that spigot. Take and clean it. And then flush it again for another minute, 30 seconds to a minute, and then take your sample. Now fill the space up, of course, like fill the bottle up to either whatever the line is that the lab, if there's a lab, the lab gives you a bottle, fill that line up, or up to the neck of the bottle, which we use other types of bottles where we fill to the neck, but it's enough to homogenize a sample. This is one of the most disappointing things we get when we get samples from the lab from other folks, is that the bottles are in disarray inside the cooler, or they've shipped in a cardboard box, which is unacceptable because it just leaks air, it leaks water, and it's not great. We've gotten samples in buckets, and I've seen guys take their sample, wrap it up, and throw it in the side of the truck and go on to the next sample point. The sample should always be upright in the cooler, and they should be maintained all the way to the lab like that. And while I'm not recommending this for growers, we always include a temperature bottle, which is a referent bottle, what they call a referent bottle. It's so that you've maintained that cold chain all the way to the lab. This is what it should look like. That's what proper handling of samples look like. So getting your samples to the lab, I'm not going to go into this graph too much, but this is a whole time study we did. Basically, when you take a sample and you get it to the lab, what happens to that bacteria in that sample? And as you see, this is the regulatory threshold, the EPA method threshold. So 6 hours to get it to the lab, 2 hours for the lab to process it. Right there, the orange line is the 2 hour, and you see a pretty steep drop in these samples that are held for longer than the regulatory threshold. So it's important to, once you take the sample, get it to the lab as soon as possible. We recommend you use a chain of custody. We have these available, and we gladly hand them to the CCOF if they want to distribute them if they get asked. We use these for everything. That way, once you take a sample, you ensure that you've maintained that custody and you've given that custody up to the lab. And I think labs, I know labs actually provide these, and they're required when you turn in samples to a lab. Okay, I think we have a quiz here.
Yeah, that's right. So we're gonna give you all participants a little chance to test out your brains and see how well you were listening. So if you want to take a really good look at the photo on the left there with the person in the dark, looks like fleece, working on some water samples there, and just jot down the things you think that might be wrong with that picture. So of all of the best practice recommendations that Ronnie was talking about earlier, what things do you see that might be wrong? So I'll give you a little chance to think about that. And while we're doing that, Ronnie, I just have one quick question for you. Someone was wondering why you need to put samples on ice.
Well, just like the hold time requirement, if you don't maintain a chill, the method all microbiological samples need to be chilled as soon as they're collected, because you have a chance of either growth or death of bacteria. So they die pretty quickly if you don't do anything. So what our hold time studies have shown, and all hold time studies have shown, that if you maintain them at that 4 degrees to 10 degrees Celsius, you maintain the samples all the way to the lab there, you don't get a steep drop at all. So I think that answers the question.
Yeah, I think that's good. Thanks. All right, so we're gonna run the quiz now. So the quiz is gonna pop up on your screen, and you can choose as many answers as you want. So if you think that there's 2 things that are wrong with it, go ahead and click those 2 things. If you think there are just 1, you're not sure, click unsure. And also for those of you who have downloaded the PowerPoint slides, no cheating, because you may know the answers. So we'll take a few minutes and you can fill that out. And then we'll have little more time for people to send in their votes, or I guess answers. On GoToWebinar, they call it a poll, so I always get the language there mixed up. All right, so if you haven't sent in your answer, do it now. And I'm going to close the poll in just a sec. All right. So let's see what people thought. Maybe take a look at that, Ronnie, and then we'll go back to the picture and we can see. I think a lot of people noticed the no gloves, and there was some people also noted everything except unsure, which is good. That means we're doing a good job here with our presentation. All right, so Ronnie, I will hand things back to you if you want to continue and see how we did on our answers.
Sure, thanks, Meaghan. So you did great, actually, because yeah, I'm not wearing gloves. These are both me, by the way. I'm not wearing gloves in that picture. Another thing is that I took a surface sample. So plunging that neck down into the water column, you ensure that you will not get surface debris into your sample. So make sure that you plunge that neck down at least 6 inches below the surface and come up just like that, so you avoid getting all that surface debris. And I'm actually labeling after I took the sample. That's a big no-no. Headspace, I would argue, is probably pretty good there. You could probably lose a lot more of that. That's a 1,000 mil, it's a 1 liter sample. So I could probably lose a little bit of that, especially with all that debris in there. On the other picture, again, no gloves and my bare hand inside the cap. If you didn't notice that one is a big no-no. So taking a sample like that is a no-no. Okay, so to wrap up the practical sampling, so what are some misconceptions that I've heard over time about water sampling? One is composite sampling is better. There's 2 types of water sampling. There are multiple types, but the 2 main ones, especially for microbial water quality, are grab and composite. You may have heard of that. Take a grab sample, it's a single sample. A composite sample are multiple samples. So that means if you look at the picture there, if I took that sample and then took another and then took another and then took another and then combine those and brought them to the lab, that's a composite sample. That's not necessary. And in fact, the method, most EPA methods don't say never sample composite. Composite, I don't know if there's an adverb for that, but never take composite samples for bacterial analysis. It's too variable. The other one is that you need 2 containers, one to collect the sample and another to transfer into. That's not true either. You'll want to reduce the amount of risk of contamination, so just take it from one sample. I've heard all over the place that people are grabbing with a bucket and pouring into a sterile bottle and then taking that to the lab. That's not necessary. Just take it from the sterile bottle. The other one is that you need to use sterile gloves. Not necessary either. And in fact, the FDA uses sterile gloves when they come out and do their emergency response. If there's an outbreak or something like that, it takes 2 people. You need 2 people to put on sterile gloves correctly. Another one is that there needs to be a clean table nearby. You don't need a clean table to sample. You see me right there. I'm taking that sample. It's going right to the truck, right into the cooler. Another one was weird. I heard this thing that I put my face over the sample in that last illustration where I'm plunging that bottle down. Yeah, nowhere in anywhere does it say that you can't put your face over a sample. And another one is you should fill the bottle all the way up. Again, you know, headspace, you guys are all experts now. You need proper headspace, so don't fill that bottle all the way up. And another quick one is wet ice only. No, you can use blue ice. Blue ice is very effective. What I mean by blue ice is things that you freeze, those little blue packets or whatever that you freeze over the night and then you throw in your cooler. Those are really great at doing that, at keeping samples cool and maintained.
All right, great. Thank you. So we had quite a number of questions come in, so we'll try to get to as many as possible. And kind of going back to the FSMA requirements section, someone wrote in wondering, in a propagation greenhouse, how often do I need to, do you need to, or how many times do you need to test? Or how many times do you recommend, or how often do you recommend that the water be tested for a propagation greenhouse? Okay,
if I understand that, well, it depends on the source of water for that, and it depends on how they're irrigating. If a greenhouse, if they're overhead, if I have this correct, because I may not be clear on the practice itself, maybe, but if it depends on the source of water. If it's well source, then it's 4 times initially and then 1 time a year after that. If it's a surface water source, it's 20 times, and then 5 times a year after that as close to harvest. So it depends on a lot of things. One, the cycle, the source of water, and on how you actually, or when you actually test, or I think that's it.
Okay, great, thanks. And I think Jacob had some.
Oh yeah, it's just considering the fact that it's a propagation greenhouse and not potentially a greenhouse that's growing crops for human consumption. If it's starting the plant, I'm wondering if that falls under the rule.
No.
Okay. So that would be something else to consider too, is what you're growing subject to the produce safety rule?
No, because those are usually no, they wouldn't have to.
Okay. Although I have heard people that if you're like growing basil for public, that then gets sold at farmers markets as plants, there might be some gray area. So just, be clear on that before you go. And then another question that came in is, if you have 2 different irrigation water sources, do you need to have a water quality profile for each?
Yep. You have 2 different, well, it depends. If you're using both of them at harvest for different crops, then yes.
Okay, great.
Or if you're using both of them for the same crop, yes. If you're using them near harvest.
Okay. And then there are some quick tests for E. coli on the market. Do you think these would be safe to use for reference?
So the FDA gives you a large latitude to actually do your own sort of citizen science. So you can sort of test your way out of, if you find an equivalent method, which they did, you see that equivalent to method 1603, then by all means. But you have to have some scientific data that shows that they're equivalent.
Okay, thanks. And then also we have some growers online from Mexico that are wondering, does this rule apply to them or should they follow local regulations?
Well, right now, the water requirements are in flux, like I said, so they're simplifying the rule. We do know that, or at least we're quietly assured that there will be some testing requirement. Now, what that looks like, we won't know. But right now, this is the rule, and this is how we're training. Everyone's training on it. So, or we're talking about it. As far as Mexico is concerned, I know that they have to comply with FSMA regulation, and so I would assume yes, that they need to start thinking about their water systems and getting them sort of in line, or at least inspecting them and making sure that all these sort of regulations match up. So they won't be, yeah, there's the foreign verification program also. So maybe getting with your Department of Ag and asking them, because they'll actually be able to tell you a lot more than I would. But I would assume you would have to comply to sell to especially if you're selling within the U.S. market.
Great, thanks, that's good. So we're gonna move on to the next section of the presentation. And just to note, Ronnie, we're at 1/4 past the hour now. And we'll try to get some more of your questions in the final question and answer section. So keep writing them in and we'll try to get to as many as we can. So thanks. So Ronnie, back.
Okay, thank you. Okay, we're going to briefly talk about another provision in FSMA that requires you to sample, I'm sorry, it's not sample anymore, to conduct an environmental assessment or an annual inspection. So we'll get into that right now. And if you haven't started these, it's a good time to start now. And what we mean by environmental assessment is you need to understand the nature of each water source. Now, this is language from the rule itself. The nature of each of your water sources. So you know where your source is. Now you need to inspect around it and understand the nature of it. For example, is it groundwater or is it surface water? It's a pretty easy answer, or a pretty easy question to answer. The extent to which it's under your control. So how much control do you have of that water system? And what is the degree of protection for each of these water sources? And you also need to understand who your neighbors are. What are the adjacent land uses nearby? Do you have a CAFO nearby? Well, you need to note that in your environmental assessment. And so in the law, it says you need to understand the likelihood of an introduction of a known or reasonably foreseeable hazard to the agricultural water by another user. Again, getting to know your neighbors and understanding what happens to that water before it reaches your farm. You should understand that. If you don't already, it's a good idea to get started doing that. So when do you need to do this? Well, they recommend that you do it at the beginning of the growing season or as appropriate. So different the growing season could mean anything. You could have multiple starts to growing through the whole season, but they say in the beginning, get to know your water source. And get to know, so specifically, what do you need to know? So you need to know what are your upstream inputs. So what is going into the stream? Do you have confluences like this? Is that going on? You have a turbidity, a high turbidity confluence going on? You need to know that. Do you have livestock operations nearby, near your water? Do you have wildlife? Wildlife inputs are pretty common. You'll see this along canal roads all the time. I captured this probably 3 weeks ago, or 4 weeks ago in California. So this happens all the time, and these just slip off right into. You get driving down the road and they just go right into the canal. So you need to understand that, need to note that. Are cattle's loafing in your water? Not your water, in the water that you're bringing onto your property? Are they loafing upstream in the waterway? What happens? And is there canal maintenance going on? Is there an event that happened, like this limb falling into the canal that created a pretty big turbidity plume as it was being removed? So you'll want to understand that if you can. We developed at UC Davis, we developed a pretty simplified annual inspection form. It's a 2-page form. It covers basically all sources of water that go onto farm, and it's that includes surface, ground, and pipe water, and that includes ponds and things like that. It's also tablet and binder ready, meaning it's an interactive PDF, or it's printable, and you can fill it out and then put it in your binder. And we completed some instructional videos to go along with that, how to take a sample. So everything I'm talking about today, we're actually going to put on our website on how to actually do visually versus the webinar where you just hear my voice. So what do we mean by source inspection? Well, in a piped water system, you'll want to look for things like leaks, breaks. This leak in particular they had heavy biofilming going on, and it was actually reaching up into the pipe system. So it was contaminating the water. So we recommend that you clear up leaks and any breaks going on. Now, backflow prevention is a big subject within the FDA. When they do these inspections, like I said earlier, they look for that you have backflow preventers in place, and what we mean by that is like an air gap. With this standpipe here, there's plenty of air gap there. There's actually a calculation the NRCS uses for proper air gap. You'll want to follow that guideline. You'll want something like these pressure type vacuum breakers. You need breakers in place or double check valves, dual check valves. What's not is a valve. A valve is not going to protect your water. It can fail and go the other way. A single line check valve, I've heard this all the time that, well, there's a single line check valve in there. Well, if it fails, you have no backup. So a dual check valve is the minimum. Not required, but they'll be looking for. They call them cross connections in the rule. Backflow prevention and cross connections are the same thing. So with well systems, you'll want to make sure that your cap and casing are intact and that your well information is clearly visible. If you don't have that, try and look for it. Go back in the stack of papers and find that well information, because it's useful to have in your annual inspection of when it was drilled and all that. And then again, you want to make sure that you have no cross connections you'll want to have backflow prevention in place. Now, for surface water systems, usually at the turnout, they're usually just a head gate where a ditch rider comes in and opens it up, and you'll never think twice about it. But it's good to note if it's clean, if it's free of debris. You'll want to free it of debris if they're in place. Like this one here, this box screen is pretty. It has a lot of algae. You'll want to clear that algae out of there because they can biofilm and encourage bacteria to grow. So you'll want to clear that out before you take a sample there. And again, getting to know your neighbors, knowing these adjacent land uses. So do you have a dairy next to your pond? This is a pond, not a canal on the left. Getting to know them, just noting. You don't have to do anything about them. You just have to note that they're there, because that is a possible risk to the water that's being delivered to your property. And also, you have a neighbor that is discharging irrigation water into the canal. You'll just want to note that is occurring. So because if you don't write it down, it didn't happen. So you don't want the inspections to find that. You'll want to already be ahead of the game. So here's an example of research we did around a dairy. WA68, if you look at this green, that's a main canal. This canal is going, flowing past the dairy. And how we usually do this, we're usually in a room of people. So we say, well, what do you think? Does bacteria increase as you run down the canal from W68 to WA58? Well, it does. So that's worth noting in your environmental assessment. If you're taking samples at WA58, you'll want to note that I have a dairy adjacent to me. Now, the canal up above is, is a lateral canal, a smaller canal, WA69, 71, and 70. If you look at those, do you think that they're actually influenced? Well, they are. They are as well. So if you have any sample, if you have samples at the end of this canal, you'll want to know that you're in the influence. Those red lines are prevailing winds, and they actually have an influence on the water up there in that lateral canal. So how can we help, or what are we doing currently right now as far as FSMA is concerned? Well, we're looking at cooperative monitoring, a small provision in FSMA where the FDA said growers on a shared source, so you're along a stretch of canal, can defensively sample together. So one guy takes a sample or one gal takes a sample, and that works as data for all the growers in that sort of stretch of canal. So we're looking at that now. We just finished that sampling up this year or in last month, And what it says is that you can do this as long as there's no reasonably identifiable contribution between sample points. So if you don't have any point sources like a dairy discharge or sewage or any confluences, this is pretty much you can do it, but it doesn't specify at what scale. They do say that at a watershed scale, it's probably not possible, or at a large river system, it's probably not possible, but it could be. You could test your way into it, especially if you know your neighbors and you guys all kind of collaborate with one another. So you basically are sharing the risk for that. So we're looking into that. We're going to start publishing on that. The FDA is keen on getting this out there because it is a cost sharing proposal or project. So it would be super beneficial to growers if they could actually share data. So what we are seeing is that short stretches, and this is preliminary results, short stretches they can defensively sample together. And growers on pipe laterals, same thing. If you have a 2-mile pipe lateral or 5-mile pipe lateral, 5-kilometer pipe lateral, you can defensively to share data. Even growers without a point source, like I said before, you have a large stretch, no point source, they're actually able to cooperatively monitor. And it doesn't mean that growers with livestock operations can't. It just means you have to be able to show that that it's the risk is that the variability is there and it won't affect your water quality profile. I'll explain more about that here in a little bit in the next slide. So what does variability mean? Well, it's not a bad thing. People think that if I get 100 CFU and then tomorrow I'm at 20 CFU and then I'm back up to 100 and it stays 100, that's a bad thing. It's actually not very bad. That's actually pretty low. 126 consistently at the threshold would be bad for criteria. But if you know your water source, you know your water source. You know that at some point the variability will keep you in line. But you need to understand what makes your system unique. So understanding your water delivery system, understanding how the water is conveyed to your farm, and any inputs, again, getting back to that environmental assessment, and is a good way to understand that variability. You get a spike in up to 100, and then you're down to 20, and then you get 100, and then 20. You need to understand what's going on that makes it go that way. So getting to know your neighbors also, not knocking on doors, but kind of looking upstream and seeing what's going on. FSMA states that you can sample more. You don't have to stick to the 20 samples. You can go 40 samples to understand your variability. We know that when we sample, if we get a high count, we return and we try to figure out where that's going. We take more samples upstream to see if we can get a point source, something that we can point to that is a source of that contamination. But this is not required by the FDA or by FSMA. Okay. Any questions?
All right, thank you. That was a nice overview on environmental assessments. And yes, we have more questions came in. We have a couple of growers that are either on municipal water or municipal reclaimed water and are wondering how frequently they would need to test those water sources.
Right now, municipal water is, as long as you have tests from your, whoever the utility is, delivering that water to you, they're zero detect. You don't have to sample yourself. They're doing it for you. So just get that get those results from them.
Okay, great. And that would be the same for reclaimed water as non-reclaimed water?
Yes, because it's treated.
Okay.
It's, yeah. Well, reclaimed water depends on, well, it depends. You just have to figure out from the utility if they're testing for E. coli. If they are, then they probably are. So I would say get the test from them to understand it.
Okay, that sounds good. And make sure that they're testing for what FSMA requires.
Yeah, because purple valve water, I don't know how far it's treated if it's like that, but yeah, get to know. I think you can talk to I'm blanking on it, but yeah, just talk to your utility. Sorry.
Okay, great, thanks. So I just wanted to be conscious of time and note that it's 1:30 now. For those of you that need to jump off now, we do have a recording that will be sent out later, so you can listen to the final questions and answers. But I wanted to have a little more time to ask Ronnie some more questions, so we're going to run over just a couple minutes. So we also had a question that came in asking, is there an amount of time between irrigation and harvest that would eliminate the requirement to test irrigation water?
Yes. And it depends on when last harvest is. So they have the metric for that in the rule. But I think it's like I can't remember the actual which one it is. They have a microbial die-off metric for that. So if you go to the rule and look for microbial die-off, just type that in the Federal Register, and they give you tons of comments on that, on how to actually. You still have to test, of course, when you need to test. And maybe I didn't understand the question. Can you repeat the question again? Sorry, I may have gone on a tangent.
Sure. So is there amount of time between irrigation and harvest that would eliminate the requirement to test irrigation water? So like if you were irrigating once and then 6 months later you're harvesting the crop or something like that?
No. Especially if it's touching the harvestable portion.
Okay, so it sounds like you need to test no matter what if you're using irrigation water.
Yep.
Okay, all right, good.
Yeah, and I'll just add that the FDA builds in a half a log die-off per day. So you can plug these into various calculators. University of Arizona has one, I believe, and you plug in your water test, and that'll give you the amount of days that you can. It automatically calculates this log, 0.5 log per day die-off, and then you'll know how many days you can wait in between, depending on what your water samples are reading, how many days you can wait between harvesting and irrigating your crops. So hope that helps.
Great, thanks.
Yeah, we have on our water quality profile, we also have that same spreadsheet. We also have a die-off calculator.
Great. So we had another question come in wanting to know if the water tests have to be reported in numerical values, or can they be presence-absence reports?
Presence-absence is only available for post-harvest water. Municipal sources, or not even municipal sources, post-harvest water can be, they have some presence-absence. They just released more methods that use that, but for pre-harvest water, nothing in agricultural water that you can do a presence-absence test. Okay,
great, thanks. And then just have a couple quick questions kind of as to, does something need to comply or not with the water rules? So if you had furrow irrigated broccoli, is that something that would need to comply with the production water requirements?
That's a maybe. It is incumbent upon you, the grower, to make sure that water isn't coming in contact. So if it doesn't, and you know it doesn't, then you don't have to comply, or you probably don't have to comply. I'm not going to say a definite no, because things are different. I said the same thing about tomatoes. I had to take that out of my talk, but I know that tomatoes tree out, or they bush out, and some of them actually get into the furrow. So yeah, broccoli not so much. So I would say no. I'd say you wouldn't have to comply.
Okay.
I might consider it a yes, because some people eat the stalks of the broccoli, but I guess you could look at it a couple different ways.
So it sounds like it depends on your system and what you're selling and a bunch of different factors.
Yeah, it's context stuff. Yeah.
Yeah.
Yeah, but and some people, and again, farms are super complicated. If you're spraying with surface water, any kind of either fertigation, chemical, whatever it is, then, and you're not, conveying that in, you're not going to do it in the furrow, then yeah, you would have to comply. So there are other circumstances. In and of itself, furrow irrigation doesn't necessarily trigger a compliance.
All right, great, thanks. And then just one last quick question. Someone was wondering if potatoes are subject to the produce safety rule.
Not as of now. Nope.
All right. Great. So I think we're going to wrap things up. Thank you, Ronnie, for your great presentation and going over all those best practices for taking water samples and doing site assessments. I know you have a lot going on, so thanks for joining us this afternoon. And yeah, I'm just going to go over a few more resources. Ronnie mentioned that if you're curious and want to understand FDA's thinking around the FSMA Produce Safety Rule, to check out the rule itself. So here's a link for you to read, and Subpart E would be the water section. These are just some general on-farm food safety resources from our partner Community Alliance with Family Farmers, as well as CCOF. And if you're still trying to figure out whether you are fully subject to the FSMA Produce Safety Rule, the National Sustainable Agriculture Coalition has a great flow chart that'll help you figure that out. CCOF has some upcoming events. If you are a farm that is fully subject to the produce safety rule and are required to send a supervisor to a FSMA approved food safety training, we have a couple of those coming up in November and January. So check those out. We got a grant from the USDA to help support those, so they're only $35 right now, which is good. Non-grant supported workshops can be up in the hundreds of dollars. So we're encouraging everybody to take advantage of them right now, even if you might not need to comply. Once you get out of the way, you're good to go before the prices go back up again. Right now, everybody's really wanting to help all the growers get on board, so take advantage of that. In November, we have a tour of the San Francisco Wholesale Produce Market, and we also have partnered with the Organic Produce Network to host the Organic Grower Summit in Monterey in December. And if you have any high school students in your life, the Future Organic Farmer Grant Fund here at the CCOF Foundation is accepting applications for organic-focused FFA supervised agricultural experience projects. So send them our way. And thanks again for attending. Again, if we didn't get to your specific question, please follow up with us. We'll have some contact information in the email that goes out. And if you would like to receive Certified Crop Advisor Continuing Education Units for this webinar, please fill out your full name and CCA certification number in the evaluation form. So thank you again, and have a good afternoon.